think in here and still of the sharing of the screen. Whoops, of course I messed that up. I'm gonna move our little pictures out of the way here somewhere. How you might have to mess around with that.
Sure.
Oh, is there a minimize? All right, there we go. So lots of growing pains trying to get people back in person for stuff. So it's my great pleasure to welcome Dr. Hao Zhu to our first in-person CGIBD seminar. Actually, my first ever, because I just joined this summer. And then somehow got put in charge of this. And so, thank you for coming, and thank you for Lineberger Comprehensive Cancer Center for co-hosting this one. Dr. Zhu is an associate professor at the Children's Research Institute, UT Southwestern, where he holds the Kern-Weidenthal Distinguished Professorship in Pediatric Research. He's also an attending physician in the multidisciplinary liver cancer clinic at Parkland Memorial Hospital. So now I got all the names of things out of the way. He has an incredibly accomplished CV, and I could find only one flaw, and that's he got his bachelor's degree from Duke. and he knew I was going to make this joke, most likely, even if I didn't tell him. And then he went on to Harvard Medical School and MIT. He underwent his training in internal medicine at the University of California, San Francisco, and medical oncology at Dana-Farber. He then performed postdoctoral work at Boston's Children's, where he started working on connections among RNA biology, cancer, regeneration, and mouse models. And then in 2012, started his own lab at Children's Medical Center at UT Southwestern. His lab focuses on identifying genes and mechanisms that regulate regenerative capacity in the liver, and understanding how these contribute to hepatocellular carcinoma development. And over the last several years, I've become aware of many of his elegant papers studying normal liver biology, liver regeneration, liver diseases, and particularly roles for switch-NIF and other chromatin remodeling complexes. So they've been continuing to look at this. Many of them have turned out to be good therapeutic leads, and so I'm very excited for you to come to our talk today. Thanks, Hal.
Thanks, Justin. Well, I want to thank, you guys can hear me? I want to thank Jesse and the CGIBD for inviting me. It's really nice to be back in the Triangle. I'm super excited to be here. I'm going to tell you about mainly unpublished work, although some of it is published. Let's see here. Oh, it was working. All right, I'll just use the button. Well, it's frozen. All right, so here are my conflicts. So my lab is predominantly interested in this idea of, or this question of, why does chronic injury and tissue damage lead to tissue dysfunction and cancer? Why does it necessarily have to be that way? And, we use the liver as a model system. The liver, as you know, is the best organ. It is highly regenerative. You can cut out 70% of it. It grows back within a matter of weeks. But it does have its limitations. If you keep on injuring with hepatitis viruses, alcohol, fatty liver disease, it can lead to a chronic syndrome called cirrhosis, which is the end stage of the liver where it doesn't function very well. And along with cirrhosis comes a number of clinical phenotypes that are very problematic and difficult to treat. There's really no targeted therapeutics devoted to many of these things. You turn yellow, you build up fluid in your belly, you bleed because your vessels are large and hypertensive, and your brain doesn't function because of all the toxins that go to your brain. And if all of that wasn't enough, after all of this, you get a liver cancer called hepatocellular carcinoma. So this is a very problematic disease because when you have an organ that doesn't function and you have a cancer, the therapeutic window and index is very, very difficult to achieve in this disease. So something that my lab, as well as many other labs, are very interested in is what is going on during the storm of events. It's a very complicated series of events that occurs over decades, including damage, cell death, compensatory regeneration, and then this is associated with inflammation and fibrosis, ultimately leading to the scarred down, hardened liver, and then liver cancer. So what are the adaptive and maladaptive events on the cellular and genetic level that sort of are driving or preventing this disease from occurring? And that's what we really want to hone in on so that we can understand how to treat better. So this is a map. It's not a political map. It's a map of how common HCC is. And as you can see, it's a growing problem in the United States. Texas is a very hot area for HCC. North Carolina stays Carolina blue no matter what. But I've heard that it's a growing problem here as well. So suffice it to say, this is a growing problem in the US, even though it's predominantly been a problem in a global sense. And over the last 50 years, this is an improvement of 5-year survivals. Pancreatic cancer is down here at the bottom. The survival is really not improved, but liver is not much better. However, I'll have to say that the therapeutics for liver cancer have improved over the last 5 to 10 years with immunotherapies and combination therapies. So, we know what the common mutational drivers of liver cancer are, and those are beta-catenin, P53, TERT promoter mutations. But, beyond those, what are the factors, what are the genetic factors, the cellular factors that sort of facilitate transformation and initiation of cancer in the liver? As you can see, all of these nodules are eligible to become cancer. Why do they not all become cancer? Why is it only a few that do? What are the things that allow this transformation event to occur? And so I started my lab, and I actually did this in my postdoc. We worked on a family of RNA binding proteins called the Lin28 family of RNA binding proteins. So we started this work in George Bailey's lab, and it's, we've continued to work on this pathway. And I want to share with you our latest results on Lin28 biology in the liver. So the predominant functionality of the Lin28 proteins are two paralogs, Lin28A and B. They're originally discovered in worms as heterochronic factors that regulate the timing of developmental events. So it was discovered that Lin28s block the maturation of the Let7 microRNA family, which is a huge microRNA family, more than 10 members, and they're thought to be tumor suppressive microRNAs. So when Lin28 blocks the maturation, Let7 cannot be loaded onto Argonaute and function to suppress oncogenic gene expression. Lin28 also has another function, which is to regulate the translation of proteins through direct mRNA interactions. So it can promote translation and it can inhibit translation. And this area of Lin28 mechanism is really not that well understood. All right, so Lin28B and Lin28A are both upregulated in a fraction of liver cancers, and they're associated with poor survival in HCC. So what do we know about Lin28? So a lot of work has been done in the liver cancer space, but I'll tell you about some things that are unknown as well. So I've already mentioned it's reactivated in approximately 15 to 30% of liver cancers. It's considered an oncofetal protein because it's up in the fetus and the embryo, then it's down in adults, and then it becomes reactivated in cancer. So that makes it an attractive therapeutic target because we really don't need Lin28 in the adult tissues. We had shown before that Lin28B is required for the development of MYC-induced pediatric hepatoblastomas. So hepatoblastomas are a sort of fetal tumor that are very common in kids, or not common, but they're more common in the pediatric population. So that's something that we had discovered before, and mainly operating through Let7 suppressive mechanisms. So what is really unknown is, are Lin28s required in adult HCCs arising from a cirrhotic background? And are there non-Let7 based mechanisms for Lin28 in liver cancer? Overall, what is the therapeutic relevance of this pathway? So a while back, a graduate student, Liam Nguyen, in my lab, had found that in a common liver cancer model where you give diethylnitrosamine, this is a mutagen that causes liver cancer, you give diethylnitrosamine at 2 to 4 weeks of age, and after about 6 to 10 months, you develop multifocal liver tumors. And we had seen some low levels of Lin28B expression within these tumors. So he did an experiment where he gave wild-type mice diethylnitrosamine. You get little bits of tumor at around 6 months of age. When you do it on a P53 deficient background, you get tons of multifocal tumors. And then when he deleted Lin28A and B, you got a suppression of tumorigenesis back down to kind of wild type levels. This is pretty remarkable. It actually maybe even lower than the wild type levels. So it really mitigated the tumor initiation from DEN. Now we wanted to also overlay chronic injury. So we did the mutagen model with DEN, as well as gave carbon tetrachloride in another model where we inducibly deleted Lin28A and B in the entire body. So this is something where we wanted to mimic a therapeutic against both Lin28s in this setting. So this included chronic mutagen, chronic injury, and whole body deletion of Lin28. And so again, we got a similar result, which is that the wild type uninduced animals got tons of tumors, and the knockouts really were quite protected from tumor genesis. Only 1 out of 9 mice got tumors, and predominantly only 1 got it. And then all the wild type, most of the wild type animals got a number of tumors. So this suggests that Lin28 is important in this kind of chronic injury-induced HCC. So these are long-term models, very difficult to study. So we wanted to understand if there are kind of genetic bases for these cancers. So we had previously sequenced these DEN tumors, and we'd found that they're predominantly driven by EGFR activating mutations, HRAS, BRAF. So the RAS MAP kinase pathway is an important pathway that's driving these tumors in the mouse. So we use a trick we can do in the mouse liver cancer modeling setting, which is called hydrodynamic tail vein injection of transposons. So you can take wild type mice and you can inject these transposons containing oncogenes into the tail vein. You inject like 1 or 2 mils of this stuff along with a transposase. Those transposons will integrate into the hepatocytes and lead to tumorigenesis. So we use an NRAS G12V activating mutation overexpression model with a transposase, put them into P53 liver knockout mice. And after just about 6 to 8 weeks, you get this massive multifocal tumorigenesis. So it's a much more rapid, facile model for what we're trying to do. When we looked at Lin28 expression in these models, you did see spotty Lin28A and Lin28B expression, but not really expression throughout every tissue. So this could either mean that it was important, maybe important for initiation and development, but not for maintenance. This is something that we needed to understand. All right, so this made us look at the very earliest lesions after the hydrodynamic transfection. So we could look as early as 10 to 12 days after the injections to look at the early tumor lesions. And you notice that there's these histologically transforming lesions around the central vein, which is where most of the transposons end up going. And interestingly, Lin28 was not expressed on the protein level, but Lin28B was expressed to a certain degree within these little lesions. And IMP3 or IGFBP3 is expressed to a small degree, and IMP3 is a target of Lin28. So we wanted to make sure that this was right. We used positive controls for Lin28A and B, which are testes tissue, which express this oncofetal protein. In the double knockouts, you really see no expression of Lin28. And again, you see Lin28B, but not Lin28A in these early lesions. So we decided to test the requirement of Lin28 in this cancer model, and we got a very strong result, which is that when you gave I showed you this already, when you gave the NRAS to P53 deficient livers, you got dramatic tumorigenesis. But in the setting of Lin28A and B deficiency, you got this amazing, clean, cannot generate tumors. And we've done this more than 20 mice, and we never see any tumors develop in these. So this is a very striking, at least a tumor initiation requirement for either Lin28A or B. And again, this is showing that we can successfully induce NRAS expression in some cells within the knockout livers and the wild type livers, but you never see Lin28B because we've deleted Lin28B in these mice. Okay, so is this a model specific effect? Is it NRAS specific? We've done also beta-catenin plus YAP, strong oncogenic drivers in the liver. And again, you get a you still see some cancers here, but you get a pretty dramatic reduction in tumorigenesis upon deletion of Lin28A and B. Okay, so I've shown you this result, and the cool thing about this model is that now you can overlay with additional transposon injections to try to rescue the tumorigenesis that occurs here. So we wanted to figure out, is it Lin28A or Lin28B that's more important for this tumor model? And so when we add back Lin28B by co-injecting Lin28B with NRAS, we can rescue tumorigenesis, okay? However, when we add back Lin28A, we cannot rescue tumorigenesis, really suggesting that Lin28B is the key driving factor here. However, when we knock out just Lin28A or B, both seem to have a little, Lin28B has a more dramatic reduction in tumorigenesis, but Lin28A also has a little bit of a reduction. So we think that there may be a little bit of redundancy, but both are playing some sort of role. All right. And I'm showing you that here. So when we knock out Lin28A, you see a little bit of tumorigenesis, but there's still a prevention of a lot of the tumors. In Lin28B, you see more complete prevention of cancer. Okay, so mechanistically, we're trying to figure out what is going on, what is driving this Lin28 dependency. And so we took a series of established data sets. So we used Lin28B clip, which is cross-linking IP between RNA binding proteins and their RNA targets. We use Lin28B, Lin28A data, as well as co-IPs for Lin28 with Lin28 proteomics to look for proteins and RNAs that are interacting with either Lin28A or B. And because there are large sets of genes that are interacting, we took the narrowest possible overlap between all of the data sets. And we found a list of about 15 genes, proteins that were interacting with Lin28 on both the protein and the RNA level. And remarkably, yes, no, these from different cancer cell lines, ESLs as well. So leukemia, I think one liver cancer cell line, and ESL. So sort of a collection of different data sets. So remarkably, 14 out of these genes are RNA binding or ribosomal proteins. Okay. So suggesting that there's some ribonuclear protein complex that are interacting with Lin28 that might be important for its function. The other thing that's interesting is that most of these genes, if not all of them, are overexpressed in liver cancer. So in HCC from the TCGA, we could see that they're potentially oncogenic because they're overexpressed in liver cancer. The other thing I want to note is that within this set of genes, only one of them has canonical LET7 binding sequences, and that's IGF2BP1. So this is a known LET7 target, but none of the other genes have any LET7 binding sequences, suggesting that we're looking for a mechanism here. We're looking at a mechanism that does not have anything to do with a canonical Lin28 blocking LET7, et cetera. All right, so we had a number of hypotheses for what was going on. So it could be, and this is reported previously, that Lin28 could be increasing the transcription of some of these target genes. It could be that they're stabilizing the target mRNA levels, or it could be that there's an enhancement of translation. And so there have been examples for some of these mechanisms with Lin28 in the past. And so we wanted to sort of assess all of these possibilities. So one of the things we did was we took two liver cancer cell lines, one with high levels of Lin28 and one with no Lin28 or little Lin28. And in the high Lin28, which is H2H7, we knocked down Lin28B and found that there were no real transcriptional differences, or at least RNA level differences within the target genes that we're looking at. And we also overexpressed Lin28 and found not many significant differences in the RNA levels. So we don't think that there's a major effect on transcription. Similarly, when we knocked out Lin28, then gave actinomycin B to figure out if RNA stability, we poison transcription and then figure out if there's a stability change, and we see really no differences in the amount of RNAs after poisoning transcription. Furthermore, we overexpress Lin28 and find that there's no differences in the kind of RNA stability in this context. And finally, we assessed translational efficiency by doing polysome profiling. And so we used H2H7 knockdown Lin28. We see that there's a reduction in the polysome abundance within these cells. And we assessed what's the transcripts in the polysomes and looked at the targets that we previously identified using the genomic methods. And we found that for predominant number of these targets, the target mRNAs are shifted from the polysomes, are reduced in the polysomes when you knock down Lin28, and they're more often contained in the monosomal populations and not in the polysomes. So this suggests that when these targets are moved away from the polysomes, that their translational efficiency is reduced when you knock down Lin28. However, not all of the targets seem to have a shift in the polysomes. So it does suggest that it's not just a global reduction in translation, but there's some selectivity potentially conferred by Lin28. All right, and so one of these targets we're looking at here, RPS5, which is a ribosomal protein. When we knock down Lin28, you see a reduction of RPS5 protein levels. And when you overexpress Lin28, you see an increase in RPS protein levels. And furthermore, you will recall that the transcription of RPS5 or the mRNA levels did not seem to change, suggesting that this is a post-translational or post-transcriptional modification. So to look for kind of direct interactions between Lin28 and RPS5, we set up luciferase reporter assays for RPS5. We looked for evidence from prior ECLIP data, looking for interactions between Lin28 and RPS5. We found that Lin28 bound at selected regions across the RPS5 mRNA, and these contain canonical Lin28 binding sites. So GGAG is a motif that Lin28 tends to bind to in both exon 1 and exon 2. There's not much Lin28 binding in exon 3 and 3' UTRs. And so these were used as control reporters for Lin28 interactions. So we gave these reporters and then either added or subtracted Lin28 in the reporter assays, and then evaluated the amount of luciferase signal that you got, which is an indicator of how much translation that you're producing when you attach Lin28 binding sequences. So I'm showing you data for HU87, which is a Lin28 high cell line. So this is a Lin28 expressing cell line. And when you give the exon 1 or exon 2 reporter, the wild type luciferase signal is stronger. When you delete the Lin28 GGAG motif, or when you mutate the motif, you get a reduction in the luciferase signal for both of these reporters. And when you use the sort of negative control reporters without any Lin28 binding sequences, you see a reduced amount of luciferase signal. Now, when you suppress Lin28 by knocking it down, you see a reduction in the difference between wild type reporter and the reporters that are missing the Lin28 binding sequences. So it doesn't completely go away, but there's a suppression in the difference between Lin28 bound reporters and non-Lin28 bound reporters, suggesting that Lin28 may be interacting with the reporters at places that are not exactly where the canonical binding sites are. We also did this in the context of SNU308, which is a cell line without much Lin28. And you can see that the reporters were not that much different from each other. However, when you added back Lin28, you could see that the wild type reporter increased luciferase signal to a greater extent than those reporters without Lin28 binding sequences. So this suggests that there's some direct sequences that are interacting with Lin28 RNA binding protein that mediate some increase in translation of RPS5. So a key question, so we can show that there's some Lin28 translational targets that are directly bound by Lin28, but what is the functional relevance of some of these targets? And that's where I think the in vivo system really comes into play. So I've shown you this system. Now what we can do is add back these targets into this in vivo transposon system, and we can ask if they rescue or do not rescue. Okay, so I've already shown you this for Lin28B, but not Lin28A. And here I'm showing you when we add back known targets of Lin28 and LAT7, IGF BP1, BP2, and BP3, these can all individually rescue tumorigenesis in this context. Now, here's a control. GFP and luciferase cannot rescue tumorigenesis. RPS5 can. And then there's a number of target genes that cannot rescue tumorigenesis. But there's also a good number of these targets that do seem to rescue tumorigenesis in the Lin28 deficient setting. So this suggests that when you transcriptionally overexpress these Lin28 target genes that are translationally regulated, that it can compensate for the lack of Lin28 for tumor initiation. Those are one at a time. Yeah, so we take each target and inject into about 5 to 7 mice, and then ask if they can rescue tumorigenesis within the same time frame. Well, so these will all compensate. Yes, yes. So that is a. So what I'm thinking is that the level of target that you're producing is highly overexpressed. So Lin28 would ordinarily increase each of these levels by a small amount, but when you increase them by a large amount, that they're somehow sufficient to rescue tumorigenesis. I think there's some more work to be done in terms of how these collection of targets might, sort of rescue the Lin28 deficiency, but it suggests that these livers are on the brink of getting tumorigenesis, and then adding back some can sort of bring it back. Yeah, that's something that we're doing now to make sure that those early tumor lesions have some either deficiency or activation of these targets that would sort of corroborate the data that we're showing here. So what I'm showing you here is that what I think is the most remarkable finding is that mice without Lin28 really have a significant impairment in tumor initiation. And at the same time, these mice are completely normal. They function normally, their livers are normal, they regenerate normally. So you're really finding a key factor that is essential for tumor formation. We have also done some studies that show, and I haven't shown you this here, but it seems that Lin28 is not required for the progression of these tumors, but it is important for the initiation. And that's consistent with the expression data that I showed you, where there's some cells that express Lin28, but not all of the cells. Lin28 directly binds to and increases the translation of many ribosomal and RNA binding proteins. And some of these can rescue tumor formation in the Lin28 deficient mice. So I think tumor initiation is often thought, or progression is often thought to be more important than initiation. But in the context of liver disease and HCC, tumor initiation is extremely important because usually you get one tumor, you cure that tumor, you get rid of it, but then there's the entire liver is eligible because of the field effect to produce more tumors. So we're constantly thinking about tumor initiation in the course of this disease. Okay, so for the second part of the talk, I'm going to focus on regeneration. And so one of the things my lab is doing is trying to look for new pathways to improve regeneration. And we use the liver as a model system, but we try to apply what we've learned in the liver maybe to other settings, because the liver does regenerate quite well. And so things that we find here, number one, might alleviate some of the issues with cirrhosis, but it might also be applied to other tissues for improvement of regeneration. And once we can find these pathways, we want to ask, does more regeneration lead to more cancer? Are they causally connected, or can they be dissociated in some way? So a key kind of technology that we've been trying to focus on in my lab is to do in vivo genetic screens. And so this is a system that was set up by a very talented graduate student, Joyce Jha. She set up transposon-based systems, and I've already introduced to you the hydrodynamic transfection-based method, where these Sleeping Beauty transposons contain an FAH rescue gene. This is an enzyme in the tyrosine metabolism pathway we can inject them into these mice with tyrosinemia, which is a genetic disease of the liver, where tyrosine metabolites are built up because they can't be catabolized. And so these mice die without a drug that rescues them. However, if you inject one of these plasmids in there, the FAH will rescue the knockout clones and then grow, because it then serves as a kind of a selection marker. We have Cas9 in here, or dead Cas9. It's a Crispr A system. We've got a guide strand that we can use here. So we can develop pools of these sleeping beauty transposons that you can co-inject with the sleeping beauty transposase into the tail vein of the liver, shotgun the liver, let them repopulate over several weeks, and then grind up the liver after that time to evaluate the guide strand composition to figure out which guides mediated increased regeneration or decreased regeneration. So we used a library from Chris Vakek's lab from Cold Spring Harbor. He made a very nice CRISPR-based library to target the functional domains of many epigenetic proteins, chromatin remodeling genes, many of which you guys are familiar with. So we thought that this is a perfect size library. It's about 160 genes. We did 5 or 6 guides each, so it's about 1,000 guide library that we could put into the mice. And we did both CRISPR knockout and CRISPR A screening. So our expectation was that if genes are showing up in both of these orthogonal libraries, then they become very strong hits for regeneration. So one such family of genes, BAS2A and BAS2B, showed up as regeneration inhibitors in the knockout screen. So when you delete these genes, it promotes clonal expansion and regeneration. However, when you overexpress these genes in the CRISPR A setting, it could mitigate or reduce the clonal expansion. So we felt that the fact that they show up in both screens really got us excited about the validity of these genes in the regenerative process. And there's many other genes in here that we didn't focus on because of various reasons, and some of which show up in both screens as well. So you guys are super familiar with the chromatin remodeling complexes. There's many different ones. The most famous one probably being sweet-SNF, giant complex, uses the energy of ATP to move nucleosomes. There's an imitation sweet complex, which is actually a family of complexes that include the nucleolar remodeling complex, of which contain the SMARCA A5 enzymatic component, and BAS2A and BAS2B, which are the co-regulatory components of these smaller complexes. It's nice that they're smaller because they make it a little bit easier to work with. So we looked at the expression of BAS2A and BAS2B in the context of regeneration. We used a partial hepatectomy model where you take a normal liver, cut out 70%, and then let it grow back. And we take different time points to measure gene expression. And we saw that both BAS2A and BAS2B showed a reduction of expression with a nadir at around 8 to 25 hours. And also in human brain tissue, there's a increase in BAS2B over age, suggesting that BAS2, so all of this data together suggests that BAS2A and BAS2B might be regeneration suppressing genes that go up with age. So we did joisted validation experiments with single guides against either BAS2A and BAS2B. So she again injected these transposons, looked for clonal expansion, which is marked in red here. And you can see the control has much less expansion as compared to BAS2A and BAS2B. And in fact, BAS2B had a slightly stronger effect than BAS2A in terms of promoting expansion here. She did the opposite experiment, which is to overexpress cDNAs of either BAS2A or BAS2B along with FAH, and again saw a reduction in the clonal expansion when you overexpress these constructs. So this got us excited, and we made knockout mice. The first knockout mouse that we made was a BAS2B knockout. And this is a great mouse because they look totally fine. There's no phenotype at baseline. This is a year-old mouse with no problems with any of its organs. They grow the same. Their livers look normal. So they're pretty much normal mice. However, when you try to regenerate them, they have increased regenerated liver mass at fixed time points, suggesting that what we saw in the sgRNA screen was similar to the whole body knockout mice. They have more proliferation after partial hepatectomy, and the gene expression and proliferation corresponds to the increased proliferation rate here. But if you look at mice that have not been induced to regenerate, there's no difference in cell cycle genes. So only when you activate the regeneration signal, only when you introduce injury, can BAS2 deficiency promote regeneration. So this is different than if you overexpress a cell cycle gene or MYC, you cause constant overgrowth or hypertrophy or hyperplasia. This is not that case. This is a conditional or signal-dependent driver of proliferation. So at the same time, a group in China published a Nature paper showing that BAS2 deficiency in worms could increase longevity in worms. And that was pretty cool because that was another good thing about BAS2 deficiency. And they had made BAS2B knockout mice as well. And they claimed, and I'm no neuroscientist, but they claimed that the older BAS2B deficient mice had increased cognitive function and memory in a variety of different tests. So suggesting that getting rid of this protein has certain benefits. The downsides we're not sure of yet, but there are some benefits here. The other good news is that there are bromodemain inhibitors of BAS2A and B, which are available from GSK as well as other consortia. And these are very independent scaffolds, but they're bromodemain inhibitors, and both of these show very similar effects. So inhibition of their bromodemain seem to block the accessibility of BAS to chromatin, and we've confirmed that. So what's interesting is that when you give the inhibitors, it mimics the genetic effects of BAS2 deficiency, promotes regeneration, doesn't have any effect on baseline liver function or liver growth. Also, the inhibitor becomes a very useful tool compound for toxicity studies with carbon tetrachloride. For whatever reason, there's a reduction in the amount of necrosis after carbon tetrachloride injury, and there's an increase in proliferation. Similarly, if you give BAS2 inhibitor right before a lethal dose of Tylenol, you keep the mice alive at a much higher rate than if you don't give the BAS2 inhibitor. So this is BAS2 inhibitor given right before the toxic dose, right after the toxic dose of Tylenol. So this is a common scenario where suicide attempts are made by drinking and Tylenol. They show up unconscious in the ER and they give N-acetylcysteine to try to rescue them, but it doesn't always work. So the BAS2 inhibitor could be a possible sort of therapeutic value in this situation. And again, we try to extend the results to the colon, and we use the DSS model of colitis. And you can see that the GSK inhibitor compared to the control inhibitor had some benefit in keeping the weight increase in mice undergoing colitis. Colon length was increased as well as proliferation in the crypts of the colon of mice treated with 2801. So we now have data showing that when we knock out BAS2B, there's absolutely no phenotype. When we knock out BAS2A, there is an even stronger phenotype than this. So this result is due to BAS2A deficiency and not B deficiency, suggesting in different tissues there's different requirements for BAS2A and BAS2B. All right, so what's the mechanism here? So Joyce did RNA sequencing of the regenerating livers, found a bunch of genes that had changed after 40 hours of liver regeneration. Many of them you would expect, cell cycle related genes, MYC targets, translation, mTOR signaling, differentiation, metabolism related genes are downregulated. So to try to sort out what was going on here, we tried another CRISPR screen where we gave representative genes, or we knocked out, actually CRISPR activated representative genes from these different pathways, and then either gave the control drug or the BAS2 inhibitor to figure out which of the genes might be mediating these effects. And it's a little complicated, but what we're trying to do is, let's say bastu inhibitor is driving up cell cycle related genes, then the activation of those genes would not be selected for in the setting of the bastu inhibitor, but it would be selected for in the negative control setting. So what we found here by doing this screen, some of the genes showed differential selection. And predominantly there were cell cycle genes as well as ribosomal proteins and protein synthesis related genes. So we started to look into this. And so we did, in vitro, we did bastu flag chip-seq with and without inhibitor. Sorry, without inhibitor, with inhibitor. With inhibitor. I think this is flipped. So bastu is binding 26 ribosomal proteins. With the inhibitor, the binding is no longer seen. Okay. Same thing for bastu. With the inhibitor, the chromatin is more open on these ribosomal proteins, and activated histone marks are increased in this context as well. Moreover, a predominant amount of these ribosomal proteins were upregulated in the setting of bastu inhibitor. So just because ribosomal proteins are up does not mean that protein synthesis is up. And so we try to confirm that under bastu inhibitory conditions, looking at a marker called OP-Puro, which measures the protein synthesis levels, we could see that the inhibitor increases protein synthesis globally compared to vehicle, and rapamycin decreases protein synthesis, as you would expect. This is the quantitation shown here. And furthermore, you also see an increase in 45S ribosomal RNA transcription, which is the original known function for BAS2 and imitation SWE, or the nucleolar remodeling complex, is to heterochromatinize ribosomal RNA. So when you release the brakes on ribosomal RNAs, you see an increase in ribosomal RNA production. What we're also showing that ribosomal proteins are also released when you take off BAS2 proteins. So then Joyce did an interesting experiment, which is to individually overexpress ribosomal proteins, wondering if their individual overexpression of these proteins under this FAH regeneration system could mediate regenerative functions. And what she found is that most of them didn't do much compared to control. However, 2 of them did seem to, on their own, drive regeneration. You can see that overexpression of RPL24 or 10A could increase FAH repopulation. So what we haven't done here is measure the ribosomal content or the ribosomal abundance. And we don't know if these are driving ribosomal abundance increases or doing something non-ribosomal. So that's something we don't know yet. We then asked if these proteins are essential for the BAS2 phenotype. And so we use the RPL24 mouse model, which is called the belly spot mouse. This is a heterozygous mutation in RPL24, and the mice develop normally, except for this weird tail and this belly spot, and they have trouble increasing protein synthesis. So for example, these mice do not develop MYC-induced tumors as well as wild-type mice, but they do develop P53-dependent tumors in the same way as wild-type mice. So it's a selective abrogation of protein synthesis increase. And so what I'm going to show you here is that basically the inhibitor works in wild-type mice, but does not work as well in the setting of the RPL24 deficient mice. And you can see that most clearly with cell proliferation. So these really abrogate proliferation when you lose one copy of RPL24. So the basic model here is fairly simple, which is that, under sort of normal conditions, under sort of uninjured normal quiescent conditions, having more or less ribosomes doesn't really matter. You don't see it. But when you demand regeneration, having an increased ribosomal burden at the ready seems to promote tissue regeneration in this context. All right. So in the last just 2 minutes, what I'll say is that one of the things we're really interested in is to use these in vivo screening tools to understand the somatic biology of the liver. The cirrhotic liver is so filled with different mutations and these are mutations that are not related to cancer. So this is a really important study done by the Sanger, Peter Campbell's group, who sequenced NASH cirrhotic livers. And what they found was that there are a bunch of genes mutated recurrently in the NASH liver. And they're interesting genes. They're metabolic enzymes related to lipid metabolism. So this is setting up a hypothesis that the damaged liver is mutating genes that might provide some adaptive benefit to the liver. The human livers are doing their own CRISPR screens to figure out what could help them, okay? And so I think we can use this as a way of understanding somatic genetics. So the idea is basically borrowed from cancer and borrowed from cell composition. Winner cells and loser cells tell you something about the genetic defect in these clones. And using this system not only teaches you about regeneration, but about adaptation in the face of chronic liver disease. And so without getting into it, we're essentially setting up these CRISPR screens in the setting of high-fat diets, alcohol, other human liver injuries, to understand what are the clones that arise and what are the mutations that lead to the clonal expansion. Because I think this is going to be another way of figuring out the important genetics of the liver. All right, that's basically what I wanted to say about this. So this is a future approach that I think is going to be widely applicable for many liver disease and other kind of disease models as well. All right. So I want to end by thanking the people who did the work. So the Lin-28 work was really kind of spearheaded by a postdoc who recently started his lab back in China, Yonglin Wei, as well as a graduate student, Sean Shea, who's sort of helping him wrap up the paper. The BAS2 work was done by Joyce, who set up all the screening tools and really carried out that work. Liam also helped with the Lin-28 work. And the fatty liver screening and it was done by Zishi Wang. And he's doing a series of other screens that hopefully will come out in the near future. I want to thank our funding sources and be happy to answer any questions that you guys might have. You know, my understanding, and we haven't looked in development that much, so BAS2 seems to be ubiquitously expressed in multiple tissues. But I actually am not sure if it's higher or lower in embryogenesis or adult tissues. I suspect that it's lower during embryogenesis and higher during adulthood, but I don't really have clear evidence of that. No, so no one has done anything. So the BAS2A was thought to be an oncogenic driver of prostate cancer. So this is really interesting. It seemed to have pro-oncogenic functions in prostate cancer. So what's intriguing is that, you know, so I think people were developing inhibitors thinking that it would be a good therapeutic in prostate cancer. So it's intriguing to me that you can block an oncogene and promote regeneration. But I think this is like many chromatin remodeling genes. There's a lot of context dependency. So can you really call it an oncogene or a tumor suppressor? It's hard to say.
So do we know what the other parts of that complex are?
So it's BAS2A and SMARCA5 are the core complex. It also interacts with HDACs, DNMTs, so DNA methylation factors. So there's a lot of factors that regulate the heterochromatin around ribosomal RNAs and probably ribosomal proteins. Yes.
Right. Very nice. Nice talk. So I think your the pathway you're studying really links this phenotype to the protein synthesis or protein level of ribosomal proteins. And in his lab here, number actually found that the MDM2 bind to this ribosomal protein, and therefore modulate the p53 signaling. I'm wondering if the phenotype you're studying here really kind of due to the p53 event. So in a way, how ribosomal protein really contribute to the regeneration or the other phenotype?
Yeah, we haven't looked at p53 dependency of the model. We're just starting to think about doing cancer modeling with the bastu knockout mice and using the inhibitors in different cancer contexts, which will certainly be p53 dependent in some way. But we haven't looked into that at all.
Anybody on Zoom want to ask a question? You can put them in the chat or unmute yourself.
Yeah, I have a question. Hey, very nice talk. Two things. One is, do you have any thoughts about whether the genetic background would have, if the mice would have any effect on the Lin28, the intensity or the degree of the Lin28 knockout effects on liver cancer? And related to that, the rescue with the bastu inhibitor with the Tylenol poisoning, that's another, there's some nice studies showing a wide range of response in mice to Tylenol poisoning. And I'm wondering if anyone's looked at bastu or A expression across different mouse strains.
Yeah, so the first question on Lin28, so the model, the strain background we use is FVB, which is the white mouse, has a huge pronucleus and is very cancer prone. So you inject anything into those mice and they develop massive tumors very quickly. And that's kind of why we use it. So Black 6 will develop tumors as well, but it takes much longer. So we really haven't tried in different strains. But it could be that the magnitude of the effect is reduced in something like Black 6 and amplified in something like FEB, but I don't know. For the bastu thing, I guess one thing I'll say is that it's interesting that Lin28 and bastu are both promoting translation on some level, but one is doing it in promotion of cancer and one is doing it in promotion of regeneration, to simplify things. But your question related to Tylenol, I really don't know. I know that there's a lot of studies with different strains, but I'm not familiar. And we haven't tried it in different strain backgrounds.
Thank you.
Hi.
Yes.
So for the bastu A and 2B, they have a bromodemain. Do you know what it's interacting with? What acetylation mark? Is it the same for 2A and 2B? Or they like how similar are those two proteins?
Yeah, they're very similar, but I don't know exactly what they're mediating. I'm not sure about that.
And it looked like you've done some chips. Like, are their targets identical for bastu A and 2B?
Oh, they overlap with the ribosomal proteins and a lot of growth-related genes, but there's some differences as well. So I'm not, and it's a little bit dependent on how much. So we overexpress these with a flag tag, and so the efficiency may depend on the amount of protein that we're producing. There are differences, but I think there are a lot of overlap too.
I have a question.
Yeah.
I'm thinking follow up to Shelley's question about the fact that so many
Yeah.
rescue.
Yeah.
I was wondering if you ever considered it might be that RNAs themselves are not the protein, and there's something maybe the binding sites in there. If you could transfer that in an experiment to GFP or whatever, and show that that can also rescue.
Ooh, that's a really cool idea. So the binding site of Lin28 might be mediating some sort of
Right.
protein RNA interaction or act as a sponge or something like
Right.
that. So if you overexpress the binding site,
But,
Yeah,
I'm
yeah,
trying to
yeah.
figure it out, but
yeah, we are struggling with this question of why are there so many things at rescue? And it's a very valid question. And I'm wondering if it's something about the model where it just needs an extra little bit of translation or a little component. And then when you overexpress just one, but I don't, that's an interesting idea.
just knock it out. of
But the
brain.
sponge thing doesn't make sense because, oh, you're saying knocking, yeah, making a mutant.
Yeah, make it, you just,
Right.
what if it's the RNA or something encoded in the RNA?
Yeah, yeah,
Not
or
in that
attaching,
species, but this
yeah.
species.
That's a very good idea. I think we should make some nonsense mutations in the rescue plasmids to ensure that it's not something about the, that's a good idea. But a lot of these rescues are with the cDNA only, so they don't have the UTR that would mediate some of these things. But Lin28 does bind to the coding regions of some. So we need to sort that out.
Questions? All right, thank our speaker one more time, sitting online.
Thank you so much.